It's Dr. Julius again. Welcome to Choosing Biochemistry Hub. Your go-to place to master biochemical concepts one step at a time. Today we should be looking at the electron transport chain, of which we also call the respiratory chain that occurs in the inner membrane of the mitochondria. The electron chain consists of 4 protein complexes, which we call complex 1, 2, 3, 4. From this diagram, you can see the outer membrane, the inner membrane of the mitochondria, and the space in between these two membranes, which we call the intermembranous space. And right there inside, where we have our matrix. These 4 complexes are all simply protein complexes, as well as other molecules like the ubiquinone and cytochrome C, cytochrome B, cytochrome A. All of these are parts of the electron transport chain, and they simply facilitate the transfer of electrons for the reduction of molecular oxygen to give us water. And what's the significance of this? They are able to fuel the synthesis of ATP from ADP and inorganic phosphate by the enzyme ATP synthase, also embedded in the inner mitochondrial membrane. So let's see how electrons move through this chain to facilitate the production of ATP from ADP. Now looking at the first complex, complex 1, which we call the NADH reductase or NADH dehydrogenase, the enzyme that facilitates the oxidation of NAD from TCA cycle or glycolysis, facilitate the oxidation of NAD back to NADH to continue its process. And then the electron extracted by this complex 1 is transferred to ubiquinone to become dihydroubiquinone. So complex 1, whether it does that, pumps 4 protons from the matrix to the intermembrane space. As electrons move through this chain, energy is released from these electrons. So the first electrons from here are high energy electrons. By the time they get here, they are low energy electrons. The energy in the electrons are extracted in the process and is used to pump protons from the matrix to the inner mitochondrial membrane. So 4 protons leave anytime this happens. Now, complex 2 does not receive electrons from NADH. They receive electron from FADH2. If you recall, succinate dehydrogenase enzyme of the TCA cycle is part of the electron transport chain, where it oxidizes FADH2 to FAD. And then that electron, also 2 electrons from this, are also transferred again to the same intermediate ubiquinone to become dihydroubiquinone. When this complex works, no protons are pumped out. Now at complex 3, which we call the dihydroubiquinone cytochrome C oxidoreductase enzyme, this enzyme is associated with this. Apart from this enzyme itself also contains cytochrome B, 2 forms of it. Non-heme iron sulfur is also part of this. So they collect electrons from ubiquinone, thereby reoxidizing it to its original state. And then here, there's a reduction in the cytochrome C. So it will no longer be ferric. The iron, which is part of this complex, is reduced from iron 3 to iron 2. That's what happens here. And then the last step, complex 4, the job of complex 4. Now when complex 3 acts, energy is extracted from the electrons as they travel through complex 3 again, and is used to pump protons out here, thereby building up the proton gradient, which is simply difference in proton concentration between the matrix and the intermembrane space. Complex 4 consists of is the enzyme we call cytochrome oxidase, or cytochrome C, cytochrome oxidase. Also contains the cytochromes A and A3, heme ion molecules, and also copper ions. What happens then? Electrons that are here are transferred to complex 4, so that the ion that was used ion 2 here is returned back to ion 3, and then it transfers that electron to oxygen to produce water. So it's at this point that water is produced, which is the ultimate electron acceptor in the electron transport chain. What happens to all of this proton gradient that we built up here? Remember, it's a form of energy. The proton gradient is a form of the energy that we release from the electrons as they travel through this. And so that gradient is used by the enzyme ATP synthase, also embedded in the inner mitochondrial membrane, in the synthesis of ATP. How does it do that? This enzyme is a proton gradient, is a channel, so it opens up, allows the excess proton to return back to the matrix. And because it's going down the gradient, the energy is dissipated. Instead of being released as heat, it is trapped by this enzyme to add inorganic phosphate to ADP to give us ATP. This is the major way of ATP synthesis for cellular function. So while the electrons travel here, it is called the electron transport chain. But once the proton gradient is utilized by this enzyme, it becomes oxidative phosphorylation, which is what exactly the use of energy in the electrons extracted from electron carriers like NAD and FADH2 for the synthesis of ATP by the enzyme ATP synthase. Why is this that energy is extracted in this electron as they move? It's because the complex here, in this complexes in electron transport chain, their energy levels are in a decreasing order, so that these first complexes are highly electronegative, while these ones down there are less electronegative. So as electrons move, energy is extracted from them for the production of proton gradient, which is used at the last step by ATP synthase to produce energy. So this is simply a summary of what happens in the electron transport chain. I believe I had a nice time today looking at this, and I trust that you enjoyed yourself. If you have any questions, just drop them right there. They'll be attended to. Like what we do here, follow us, share us with others who may benefit from this. And as we continue to do this together, God bless you. Until I see you again, it's Dr. Juliet. Bye for now.